Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome everybody again to another episode of our show, bringing you another really fascinating guest today who is helping to create a better tomorrow on some very unique fronts in the area of mental health. We have the honor today of being joined by Dr. Srinivas Rao, who is the Chief Scientific Officer at Atai Life Sciences, which is a clinical stage biopharmaceutical company focused on transforming the treatment of mental health disorders. They have offices in New York, London, Berlin, and their business model combines funding, technology development, scientific and regulatory expertise, all with a focus on novel psychedelic therapeutic moieties and other drugs that have differentiated safety profiles and therapeutic potential. Dr. Rao also serves as the Chief Executive Officer of an Atai portfolio company called Entheogenics, which is a computational biophysics and artificial intelligence company, ultimately working to design the next generation of psychedelic-inspired mental health drugs. Dr. Rao has over 2 decades of professional experience in the pharmaceutical and biotech industries. Prior to Atai, Dr. Rao held titles of Chief Scientific Officer, Chief Medical Officer, or CEO at companies ranging from venture-backed startups to vertically integrated, publicly traded pharmaceutical companies. Dr. Rao completed his internship in internal medicine at Yale-New Haven Hospital, received his Ph.D. in neurobiology from Yale Graduate School, his M.D. from Yale School of Medicine, and he also holds both bachelor's and master's degrees in electrical engineering from Yale College and Yale Graduate School, respectively. We're honored to have him with us today. Dr. Srinivas Rao, thank you so much for taking time to come on the show.
Ira, thank you so much for having me. Really appreciate
Yeah,
it.
it's great having you. You have a fascinating background. I would love to just start off for a couple of minutes for you to take us a little bit through the early story of yourself, if you would, and how you got from electrical engineering to medicine and ultimately into neurobiology. I think that'd be a great way to start things going.
Absolutely. So yeah, my father's a physician, so at least had some peripheral exposure to medicine all the way through my childhood. He never pushed me into medicine, and so once I got to college, I was actually thinking about either physics or engineering as my major. I ended up going into computer engineering, but really transitioned very quickly into biomedical applications, as well as neural networks. And that was the very early days of neural networks and using neural networks for modeling and neural-based processing. No convolutional stuff at that point. So it was really very early days. Again, all my work was really on biomedical applications, like an intelligent wheelchair that I put together for the VA so that they could drive things around. Someone that was wheelchair bound but wouldn't run into things or fall off stairs and stuff like that. So these are some of the early things that I did, but it became apparent pretty early on that there were 2 different languages. Physicians spoke one language, engineers spoke another language. And that kind of drove some of my interest in medicine, though it wasn't really clear that I wanted to practice at that time. Some of the work I was doing in neural networks and other places really kind of inspired this desire to go into neurobiology as well on the PhD side, and really trying to understand the circuit basis of things. Cognition, how do you model cognition in a device? Well, you need to understand what's going on in the brain. And that's really kind of what kicked things off, honestly, in terms of the MD PhD. And that's basically been that's what kind of facilitated that transition. During the course of my MD training, there were points where I was like, this is kind of fun. I think I may want to do this. Then I did my internship, and I was like, yeah, I really don't want to do this in terms of taking care of patients. Academe was also not a great fit for me because of that engineering background. Academic research really is focused on, for the most part, really understanding some of the basic mechanisms, and that's great. I am an engineer and I want to solve a problem. I mean, a real world problem. And so it was kind of a natural fit, even though I didn't know it. It was a very natural fit to then transition into the biotech space. And that's what I did pretty early on.
Excellent. Excellent. And Sri, a couple of years ago, you gave a talk at it was the 10th conference of the International Society for Affective Disorders, and it was entitled Psychiatric Drug Development's Valley of Death. And you basically pointed out these three components. One, sort of just the general valley of death concept that most biotechs go through, that biotech is hard, it's expensive, and that's always going to be a problem. But in terms of psychiatric drug development, you highlighted two other really important things. One, we were over-relying on very reductionist, sort of single therapeutic, single target approaches when you had this thing, the brain, that is an ideal domain for polypharmacological approaches. And then also de-risking, which is tough in any area, but an area that, if you look in the right spots, potentially you can find areas, and we'll get into the psychedelic domain in a little bit, where you can find data in history that points to the fact, hey, maybe there's some interesting pharmacology over here that is de-risk compared to other things. Talk a little bit about this valley of death, if you would, and how it sort of led to Atai's inception.
Yeah, so I mean, the reductionist approach makes sense in many ways, right? Because if you want to try and understand something, it makes sense to kind of take it down to a single receptor system and explore that. So it's a very reasonable approach because polypharmacy, when in the context of existing drugs, tends to involve a certain degree of serendipity, and we'll get to that when we talk about pre-existing data, essentially, de-risking data. So I get it, and it makes a lot of sense. It was challenged on a couple of different fronts. Again, part of it is the complexity of the brain, but also the inability to really model some of these conditions in animals, right? I mean, with cancer, well, with with infections, it's pretty easy in an animal, right? If you're trying to treat a staph infection or a strep infection, you can give the animal that, and then you can give them the antibiotics. And for the most part, there's pretty good predictability. You can't really ask the animal how they're feeling, right? I mean, it's just a challenging thing to do. So we have all these proxy measures. So, the combination of this reductionist approach plus the inability to really model things has really stymied the field for a long time. And that kind of leads to this other angle around serendipity and these more complex drugs. Tricyclic antidepressants and things like tetracyclics like mirtazapine have much better efficacy overall than SSRIs, some of the more specific drugs. Interesting. Don't know where, presumably some of these other mechanisms are impacting the efficacy. It's hard to really know, but presumably that's what's happening. But those compounds were basically found through serendipity. They were not found through rational drug development. And the same is really true for things like on the antipsychotic side, like clozapine. Clozapine has got insanely complicated pharmacology. It still remains one of the best antipsychotics out there. It has a lot of safety concerns, but that's a different thing. But in terms of pure efficacy, it's really considered one of the best. So those are sort of the competing tensions. And, with Atai, we basically said, let's focus on the serendipity angle to some degree. Those drugs that have, they in general have complex pharmacology, but those drugs that do in fact have existing data in terms of supporting their efficacy and of course supporting safety as well. There's two reasons a drug fails. One is inadequate efficacy, but the other one is safety. So if we've got compounds that are de-risked on both fronts, that's a good place to start in terms of drug development. And that's essentially what we've done. So all of our current pipeline assets do have extensive use history, right? So whether it's DMT or ibogaine, or etifoxine or deuterated etifoxine, deuterated mitragynine, we know that there's good safety and efficacy data that already exists. And we are then going after second generation moieties. So we've got, in the context of entheogenics, as well as other discovery efforts, we are looking at structural derivatives or completely new scaffolds. There will be some NCE risk associated with those. We recognize that. So now you're going into more of the realm of more traditional drug development, and that's fine. We are still focusing on polypharmacy to some degree. And entheogenics, one of the reasons we went there with Cyclica is that their computational chemistry platform allows for targeting multiple receptor systems at the same time. So that's the reason we went there. We wanted to encapsulate potentially all the pharmacology that gives you the benefits, hack out the pieces that may be problematic. And that's essentially what we're doing there.
Outstanding. Let's if you could, can we talk about just a couple of the examples from in terms of the poly pharmacy, and you highlighted again in this presentation, there may be functional changes, but also structural changes that can be affected as well. We had I spoke to Dr. Maliskaya a couple of years ago, working with psilocybin, and there, they had this, as you point out, there's the serotonin component, but also which has a downstream effect on brain-derived neurotropic factor. Deborah Mash on the ibogaine front, having this fascinating sort of binding to the opioid receptor, but doing neat things to serotonin and nicotinic receptors as well. Talk about a couple of the interesting polypharmacy that you're seeing with some of these compounds.
Yeah, I mean, with ibogaine, I would basically say that I don't think we have a really good sense of why it behaves the way it does. I mean, the pharmacology in general, it's not obvious why there's a psychedelic or oneirophrenic effect. I mean, it's really complicated. It's also interesting that a single release of a single carbon moiety, right, changing it from ibogaine to noribogaine, gets rid of all the psychedelic or oneirophrenic effects. So it's a really strange drug. Don't know what to make of it in terms of its pharmacology. It makes things more complicated in terms of trying to find derivative compounds, and noribogaine is being developed, MC18 is being developed, but ultimately we don't know if those compounds will have efficacy in the same way that ibogaine does. We are looking at ways of characterizing its pharmacology in a more complex manner, and then through Cyclica and other technologies to try and find other, find derivatives that may get rid of some of the cardiovascular effects. Right? We know it hits hERG. It may cause some QT prolongation. We've seen that from other publications. So we know that. We're trying to figure out ways of getting around some of those limitations. Again, keeping all the good stuff, hacking away the bad stuff, but not going as fully reductionist as one would traditionally do. So DMT, kind of similar, 5-HT1A, 5-HT2. There's some dopamine activity. It hits H1. There's lots of different types of activity in this molecule as well. How does this all impact efficacy is TBD. We know that ayahuasca has some pretty good efficacy from an antidepressant perspective. Pure DMT will get a better sense of that. I mean, there are small pharmas in the midst of a phase 2A trial. Let's see how those results pan out. We're doing our own studies with our particular formulation. So, how do these different types of pharmacology impact efficacy, the different ratios? Are there biased agonist type signals there? I mean, different moieties may impact the relative activation of different second messenger systems differently. So how does that impact efficacy? All of these things we're exploring. In the context of entheogenics, we've actually generated some really fascinating compounds. They're really high potency agonists at 5-HT2A, but they're not seemingly not psychedelic in animal models. So in animal models and rodent models, there's something called the head twitch, which it seems to align with the psychedelic effect in humans. We have things that are pure agonists, essentially, that are very specific, that do not cause head twitch. Still trying to figure out what that means. And does that, they have a neuroplastic angle. Is this one of those compounds that is non-psychedelic, but neuroplastic, and non-psychedelic type of antidepressant? I don't know. We'll get to that. We're still in the early days of some of those compounds, but very intriguing. Lots of real science here. Let's put it that
Yeah,
way.
no, it's fascinating science. I've been so intrigued by the portfolio for years, and in following some of these thought leaders that you work with. Talk a little bit about the hub and spoke model, because you made a lot of bets. You look at something like the treatment-resistant depression. You have the DMT program, salvinorin A, the ketamine enantiomer. You have the relationship with Compass on psilocybin. Talk a little bit about how this model works a little bit in the sense of, you're not going to have 20 depression product programs going on, but you have quite a bit going on here.
Yeah, so let's focus on the treatment-resistant depression piece first, then we can talk about hub and spoke more broadly.
Sure.
So why are we doing what we're doing is a really good question. We are looking at compounds with very different pharmacologies, right? So R-ketamine has a glutamatergic effect. It's probably its effects are probably mediated by glutamate. DMT, 5-HT2A, salvinorin A is a kappa opioid agonist. So these are very different.
Yep.
The reason for this is treatment-resistant depression is multifactorial, right? We don't know what the underlying pathophysiology is, other than to say that there's probably many different underlying pathophysiologies. There's many different depressions. And we don't think that one size fits all. And I've said this from the very outset. I don't think that something like a psilocybin or DMT will address everybody. There's going to be people that simply do not respond. And the phase 2B results from Compass also support that. Many people responded. Those that responded had really long-term benefits. And that's remarkable. And that's a unique piece about psilocybin. But there are other people that just didn't respond. And our conjecture is that maybe some of those folks will respond with different pharmacologies. Maybe there's a different underpinning to their disease, essentially, as opposed to the more broad syndrome of depression. Maybe there's something specific. We ultimately want to do biomarkers of some kind that allow us to predict which patient's going to respond to which drug. That's obviously been a challenging thing to do in the CNS space and the affective disorder space. We're bringing to bear many different modalities, digital biomarkers, metabolomics, quantitative EEG. So we're bringing together many different things to try and see whether there's a signal there that can give us some guidance. The ideal is that you have someone that comes in with treatment-resistant depression or depression. You do a simple test, ideally just a blood test, but maybe a blood test plus something else. And then you can get a very rapid signal of where this patient should go next. And that's going to save the doctor time, and that's important. The doctor doesn't have to have this patient not respond and then have to have them come back in and switch medications. Obviously, it's huge for the patients, right? So we're trying to reduce morbidity and, unfortunately, mortality as well in this very challenging patient population. So that's the reason that we have these different angles in terms of pharmacology. Now, psilocybin and DMT, psilocybin, we know it has efficacy. We've seen beautiful efficacy. The one caveat to it, if you will, is that it requires a long stay in the doctor's office. That's why we went forward with DMT. DMT has a very short half-life. We're using formulation technology to kind of adapt. We're looking at sort of 45 minutes-ish for a psychedelic duration. That's markedly shorter than what you see with psilocybin. We want to take that. We want to take something with that kind of a profile and slot it into the infrastructure that J&J has already established for esketamine. So that's kind of our angle there. So we view that almost as the next level. We're anticipating multiple doses, repeat dosing, that's fine. But if we can reduce the burden for the patient and the site because of the very frequent dosing of Spravato, I think that's still a massive win. So that's the depression side of things. Now, in terms of the broader hub and spoke, going back to that, part of the benefits here are that we have, particularly for companies that we brought in, where it's resource efficient, right? So we are getting access to these assets without paying for the entire, paying the entire equity value of the asset. Moreover, we have a team of founders that is dedicated to the asset, that know the asset. So that's an important element. However, these companies tend to have very small teams. there's a passionate group around it, but they may not have the full spectrum of capabilities. And that's where Atai comes in once again. We have a very broad and very capable R&D team that's been put together. It's over 50 people at this point, literally running from everything from, preclinical development, discovery chemistry, all the way to late stage clinical development, CMC, from drug substance to drug product and multiple formulations. So we have the entire gamut at the Atai level. We can kind of drag and drop and put what capabilities we need in each one of these companies at the appropriate time. So it's very efficient from that perspective, because, you don't necessarily need an entire CMC department for a phase 1, phase 2A compound. You need them at sort of specific intervals, and then they can kind of back off a little bit. And of course, as the asset starts to move through the pipeline, gets into phase 3, you need dedicated teams, I would argue. Sure. But early stage, I don't think you necessarily do. And this is a much more efficient model.
In terms of entheogenics, modifying, these structures and getting rid of side effects and so forth. When it comes to, as you were just mentioning, peeling off the visionary component of some of these molecules, you mentioned obviously DMT very short, but things like ibogaine can go all day. Are there cases that you think, based on your long experience in the sort of the neurobiology space there, what you've termed psychedelic assisted psychotherapy has its place still, or do you think we're going to totally get rid of the visionary component of these compounds? And do you get any resistance from sort of the
I think it's a very interesting question. And it depends on how one interprets the psychedelic effects, right? If you think about what a psychedelic is doing, I mean, one hypothesis, and it's one that I tend to espouse, is you've got something that's causing pronounced neuroplasticity in the context of pronounced network disruption, right? So you're changing the activity of the default mode network, the reward networks, sort of the hedonic networks. All of these things are being changed acutely because of the because of these drugs, because of the 5-HT2A activation and other receptor activation. And simultaneously, you're making the brain much more malleable, right? So basically, you're changing the nature of the connections, and that presumably is driving efficacy that's also durable. I mean, it doesn't make, the pharmacodynamics of these drugs markedly outweighs the pharmacokinetics, right? Psilocybin being a perfect example. Those that respond, the drug is gone from your system. 12 hours out, there's not much of anything hanging out at that point, but you're seeing efficacy, months out. So that implies there's structural changes that are occurring. So the network disruption in particular is going to have a manifestation. If you're changing the activity of all these things, you're not going to feel the same, right? You get this network disruption when you're asleep, and that's called dreaming, right? So that's also the same kind of thing, and that's why you get dreams that have psychedelic elements. If you think about REM sleep, I mean, they're kind of disconnected from reality, and they have certain elements that overlap with the psychedelic effect. So if you view it as a manifestation of that, then the answer would be, and you need both elements. The answer is, yeah, you probably need both. On the other hand, you may be able to take just neuroplasticity in the context of some form of training, like whether it's cognitive behavioral therapy administered by a person, or it's being delivered by digital technology, which is what we're interested in. That may be just as well. I mean, that may be fine as well, right? Because you're trying to get new patterns of activity, new behaviors, and you need some sort of training, and you need it in a state where the brain is susceptible, if you will, to learning, right? Changing habits is hard. Changing habits is very hard, as I think we can all attest. And so if a drug can facilitate that would be ideal. And I think a good example of that in many ways is MDMA. Sure. MDMA, you're not really thinking about it as a psychedelic. I mean, there can be perceptual distortions, to be sure, but really the way I view it is that the entactogenic profile allows you to get a better therapeutic alliance with yourself. The therapists in the group always kind of scoff when I say that, but it allows you to tap into some of these things that you don't want to touch otherwise. It's too scary. or it's incongruent with your view of yourself. All of these different angles kind of come up. This is what this drug is doing, and then you need to kind of work through that. You need to talk through it to some degree. And that's kind of the MAPS approach, if you will, right? It's really working through some of the challenges. So that's a perfect example, really, of drug plus therapy kind of coming together. The drug may or may not be effective on its own. I mean, if you're at a rave, I don't think it's giving you any kind of therapeutic benefit. On the other hand, the combination, because of the right context, is giving you a benefit. And we know that, again, we've seen that with their phase 3 results. So, I think there's two different models here. In the psychedelic, it could be that you need the network disruption. The network disruption manifests as, psychedelic effect. In the context of something like an MDMA, it's less prominent. It's really more about having putting the brain in the appropriate state where therapy is going to really have a profound effect that's persistent. I know that was a bit of a long-winded answer.
No, it was an awesome answer. I appreciate going into that. You were talking about the portfolio, and you have some, what I'll refer to big guns on the list here of pharmaceutical candidates, where the moieties are being derived from, and so forth. I go down the list though, and then I bump into N-acetylcysteine, which I'm familiar with in my local CVS as an over-the-counter product. But you're studying it for mild traumatic brain injury. Could you talk a little bit about this one?
Absolutely.
It seems a little out of place, but maybe I'm not.
Well, I mean, there's an opportunistic element to be sure here, and we are looking at things that are significant unmet medical needs. Certainly, mild traumatic brain injury falls into that bucket. Mild traumatic brain injury is more impactful than most people realize. I mean, there's a markedly increased risk of affective disorders and other forms of, dementia and everything else that's downstream from that. So that certainly was interesting. You're right, N-acetylcysteine is available, but N-acetylcysteine delivered orally is not particularly bioavailable to the brain. Okay. And so we are looking at an alternative delivery modality, which is intranasal.
Gotcha.
And the idea is to markedly increase the uptake there. It turns out to be more difficult than one would anticipate to get it into the brain. That's sort of the secret sauce to some degree. And the idea is that, someone comes in with a concussion, you can dose them with this product and, decrease the time to resolution of symptoms, essentially, and hopefully improve downstream, manifestations of the concussion.
Excellent. Sri, your PhD was done in the title is Elucidating the Role of Inhibition in Circuit Bases of Spatial Working Memory, and you've published a series of papers in the literature on memory. Thinking, obviously beyond sort of this basket of depression, post-traumatic stress, and so forth, some of the indications you're going after. Talk about some of the larger central nervous system degenerative disorders and potential applications down the road that you may be interested there, whether it's dementia or some of these other significant unmet medical needs.
Yeah, it's interesting. The lab that I worked in was actually focused on schizophrenia.
Okay.
And cognitive impairments associated with schizophrenia are very well characterized. In fact, they can be prodromal, right? So before you have the positive symptoms, which are the hallucinations, the voices in your head, sometimes visual hallucinations as well, you actually start seeing negative symptoms and cognitive impairments. So, for example, a kid may start falling off their curves, right? They might be a straight A student, blah, 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 and actually start to decline cognitively prior to having the first psychotic break. So this is this is a phenomenon that's been described. So part of that is an alteration of working memory. And that's really the focus. That was the focus of my work and my PhD work at a very basic science level, understanding the networks that are involved, understanding the pharmacology of those networks, which actually looked at GABA. GABA as a inhibitory neurotransmitter, as well as glutamate, as well as serotonin. In fact, I did a lot of work with 5-HT2A at that time in that context. So that was the backstory. And again, that's always been that interest in cognitive impairments associated with schizophrenia is something that's come up a few times. I've always been intrigued by that space. I was involved with the development of a compound called BL1020 a long time ago. We looked at cognitive impairment in schizophrenia. It didn't work, and that compound didn't work, unfortunately. But that, that backstory is why we brought on, in part why we brought on RL-007. The Recognify asset. There's a recognition that this is a huge problem. It's an incredibly impactful problem for those folks with schizophrenia. It's one of the important reasons that they have to stay in board and care facilities, that they can't get a job. Why they have such a, why these patients are so expensive, because the fact that they can't be gainfully employed. So anything that we can do to improve on that is something that would be fantastic from a patient and societal level. Now, to your question about expanding beyond that, I think that's a really intriguing one. So with RL-007, there's definitely, if we get a signal with schizophrenia, I think there's a number of different ways we can go. I mean, I have a personal interest in autism. I think that'd be really intriguing, but the other angle there is dementia. One of the reasons that we picked schizophrenia is that it's relatively more homogeneous than something like dementia, and even relatively more homogeneous than autism. That's why we went after that as the first indication. But I think there's other angles that we can pursue there. There's actually some interest in pursuing some of the psychedelics as well, because of some of the neuroplastic, the pro-neuroplastic efficacy of these compounds. Could you utilize that? Can you harness that in some ways to improve the cognitive profile of these patients? If not, I don't think they're necessarily going to be, disease altering, but I think that they could alter the course of the disease and maybe improve cognition in a more durable manner, kind of like what you're seeing on the affective side. So again, something that we're looking into as well.
And while we're on that topic, one other thing I just wanted to ask you about, because I had a chat about maybe a year ago with Sharon Inouye up at Harvard, who specializes in delirium, and delirium is one of those odd buckets on the side here, sort of falls under sort of the hallucinogen basket, and we know there's deliriant compounds, and there's weird antihistamines from back in the 1950s and all that bit, atropine. Any interesting things? Because that's another one of those, it gets kind of misdiagnosed, and it's, this weird state of confusion, short-term confusion. Any sort of anti-deliriant pharmacology that you might run into when you're studying these compounds? Because that's another one of those, we forget about it, but it's there, and it's a major in sort of the elderly population that we confuse sometimes with just the dementia side of the equation.
That's an interesting question, and not one I've actually spent too much time thinking about, to be honest with you. I think part of the challenge there is it's more like a positive symptom, if you will, right? The confusion and everything. So the current batch of compounds that we're developing, it's not intuitive that they would be great for that. But it's not something that I've really spent a lot of time thinking about.
That's fine. Just thought I'd ask.
I appreciate that.
Yeah. Sure. What else is hot that's coming up for 2022? Any upcoming things you want to mention? Conferences you're going to be presenting at? Places that people can listen to you, meet you face to face? Please, anything that I missed, please take the floor.
Yeah, I mean, there's a few conferences for sure. I mean, the schedule is always a little up in the air, to be honest with you. But there's certainly ACNP, the American College of Neuropharmacology meeting, which will be, I think it's either November or December. I'll certainly be at that one. A lot of investor meetings along the way that I'll be at. So Citibank is coming up. There's a few others that Wainwright, a couple of others that I'll be at. In terms of newsworthy events, we do have there's a lot of things that we're really excited about that are coming up. I mean, this perception with the R-ketamine that we sort of touched upon a little bit. We have a phase 2A readout coming up a little later this year. So that's going to be really exciting. That is a putatively non-psychedelic, non-dissociative compound with rapid-acting effects or rapid-acting efficacy. That's going to be really intriguing. So truly looking forward to that readout. Got a lot of really interesting phase 1 readouts that will be coming out as well. So GABA with their etifoxine product. we've got some pharmacodynamic endpoints there as well. So that's a SAD/MAD that will support moving forward, hopefully into a phase 2A. We've got the cures asset, which is looking at deuterated mitragynine. Mitragynine is the active moiety in kratom. Very unique. It's an opioid, but a very unique one. There's not much in the way of respiratory depression associated with it. That's a big problem with opioids, right?
Yep.
Opioid-mediated fatalities are usually as a result of respiratory depression. So we can do something about that. That's huge. So that trial does have experimental pain endpoints, and we do have a comparative element where we look in a crossover manner, looking at the analgesic efficacy of deuterated mitragynine versus oxycodone versus placebo, and looking at therapeutic index. So that readout will be later this year as well. We'll also get some data around ibogaine. Kicking off a bunch of trials as well this year, and including the DMT asset and PathBio, which is an MDMA derivative, et cetera. So there's quite a bit going on, so that in terms from a news flow perspective.
Awesome. And we will stay tuned to it all and continue to follow it. But it's fascinating how far you brought this portfolio, Sri, and I just I take my hat off to you. It's an amazing set of programs, and really wishing you the best with all of it. For everybody that is going to be listening to this particular episode across the podcast networks or watching on our YouTube channel, again, you've been listening to Dr. Srinivas Rao, Chief Scientific Officer, Atai Life Sciences, doing amazing things to transform the treatment of mental health disorders. I sure want to thank you for taking the time out of your schedule to come talk to us for a little while about these topics. Obviously, thanks for everything you're doing there at Atai and the portfolio companies. And we say on our show, thanks for helping to create a better tomorrow for many people out there. It's a great story, and appreciate your time.
Absolutely. And let me emphasize, it's a team effort. We got a fantastic team, very dedicated, that's been doing all the heavy lifting. So definitely not going to take credit for it. But yeah, I really do appreciate the time.
It's great seeing you.